So, Dr. Alfie, thank you for your time and speaking to us about your paper. First of all, how did you get into the field of research?
It was really when I was thinking about doing a PhD, actually. This was the time when I was just exploring different areas in the field, which was quite interesting at that time. And it was really cell death, so-called apoptosis, programmed cell death, which really I became very, very interested in. I was fascinated by the idea that when cells actually die, that this is highly regulated and really follows a program, yeah, doing development, but also doing stress. So cells really, or whole organisms, they induce cell death if a cell is damaged, to remove it basically. Otherwise it will cause damage to the whole organism. And that's why I also choose a lab which works on C. elegans, where actually apoptosis has been discovered. So this is really how I came into the research, using C. elegans and as well also stress response mechanism, in particular to DNA damage, so genotoxic stress basically. That's really where I came over. It really goes back to my PhD, and there's really a long-term interest now, up to now, in my own group actually.
Okay. Can you give us a brief overview of the reasons for carrying out this research and the background behind the research?
Yeah, it's what I said already, it's really about DNA damage. If you think about DNA, which is basically the blueprint of life, every information in the DNA defines the whole organism and the health of the organism. So any sort of damage will affect basically the health of an organism, or the viability of an organism. So, and cells have a lot of mechanisms to maintain the DNA and keep it basically free of any sort of damage. And cells and whole organisms have a lot of mechanisms to maintain this stability. And this is really the reason why we really try to understand this, how actually a cell can maintain its genome and so the genome stability basically. So any defects in this of the genome stability will accumulate in mutations, and these mutations can then lead to cell transformation and then to the diseases. And most prominent is cancer. Cancer, the hallmark of cancer is actually it changes its genome basically in favoring of just growing wild basically. So this is really where the motivation is coming on, to study these sort of response pathways which are activated if there's any problem with DNA damage.
Okay. And what was the relevance of the research to the Fanconi anemia and the cancer?
So as I said, there's a lot of pathways and mechanisms which are activated if there's DNA damage. So there are pathways which first of all recognize the damage and then analyze it to remove the damage, so basically repair the damage. Or another way is, if the damage cannot be repaired, basically to remove the cells via cell death, via induced apoptosis. And we know already that some of the pathways actually mutated. And Fanconi anemia is one of these diseases. It is basically a genome instability syndrome. So these particular patients, they cannot detect a special lesion of DNA repair, and this is the so-called intercell crosslink. So they cannot basically deal with this sort of damage. They cannot repair it, so they accumulate this sort of damage, and this can result in cancer. And this is exactly what these children born with this disease basically have. Very early in childhood, they develop leukemia. And the only way to treat this is basically by bone marrow transplantation. Then they can live for several other years pretty fine, but then usually in the late teens, early 20s, then they start again developing different other sorts of cancer. So it's quite devastating, this disease, because we cannot basically really, we cannot heal them, yeah, we cannot really treat them, we cannot help them out. So I think but it really highlights the importance of pathways and mechanisms to detect and repair DNA lesions and to maintain the genome stability.
And what was the significance of studying the protein RNF113 in worms?
This is come from different two sides. So what I was interested at the time to identify new proteins involved in recognizing these particular lesions, these interstrand crosslinks. So in my lab we did a so-called RNAi screen, and basically you target a whole array of different genes via RNAi, a small interference RNA, and then I asked which of them, when I knock them down or delete them, they cause sensitivity to drugs or agents which cause these interstrand crosslinks. And here we found this gene RNF113, a uctin E3 ligase. So if we knock this down, this ligase, we see increased sensitivity to interstrand cross-linking reagents. So this was from my side. Our collaborator in South Korea, Professor Ko, she identified it from a different, she came from a different angle. She found that this ligase interacts with FANCD2. FANCD2 is a Fanconi-anemia pathway protein, quite a critical one. It gets mononucleated and then somehow promotes the repair of DNA lesions. So we came from different angles. Something, this gene, this ligase, this uctin ligase is somehow involved in a DNA repair pathway, right? And this is where we started then to work together, basically. And here is where basically the motivation was coming from. So there was good evidence out there that this might be a novel player in a DNA damage response pathway.
Okay. And in a couple of sentences, can you tell us what you were hoping to gain from this research?
As I mentioned already, I think what we really wanted to get a complete picture how cells can recognize and repair lesions. And there's still a lot of genes and proteins out there in the genome. We have no idea what they're basically doing. That's why finding new players are quite important to get the complete picture of a response pathway. And this ligase RNF113 seem to be one of these candidates. And by studying this new protein in this pathway, we might even find a new player which is then maybe important in cancer biology as well, because you can think about cells which maybe don't have have high levels of DNA lesions, high rates of mutations, for example. So it would be maybe a new player also in cancer biology.
Okay. Can you give us a brief run-through of the methods you used?
The method is quite complex from different sides. We use a model organism, a multicellular organism, which is the nematode, C. elegans, which is quite an interesting organism because from the methodical point of view, so to say, it's genetically very easy to manipulate. As I mentioned, RNAi works really well, so you can basically feed or inject small interference RNA to knock down genes. And there's also a way to mutate genes. And then it's quite useful for study cell biology. The worm basically is transparent. You can really look into the worm basically, and in a living worm under a phase contrast microscope. And then we also developed a lot of techniques called immunofluorescence staining techniques, which allows us to basically mark different proteins within the worm. So combining these two, the genetics on one side and the cell biology and tools from the cell biology, allowed us to study DNA repair quite nicely. Another advantage of C. elegans is that the germline is the only proliferating organ within the worm, or the rest of the cells are not proliferating, they're basically terminally differentiated. And DNA damage is particularly harmful for cells which are dividing. Because if you can think about a cell which is in the process of dividing and you damage the DNA, it will duplicate basically the damage and the harm of the cell. So that's why proliferators say very tightly regulate cell cycle in context of DNA damage and any sort of problems during DNA replication, for example. So this is really why we choose these model organisms.
What would you say was the most significant finding of the research?
I think the most significant finding is this very specific defect in homologous recombination. What I mentioned before, that this loading from this recombinase RAD51 onto sites of DNA damage, this seemed to be very specifically for interstrand crosslinks, but not for other sorts of damage like ionizing reduced double-strand breaks. So there seems to be the specificity, I think, is quite interesting, and also tells me in a way that lesions can be very different, and every DNA lesion has a different set of responses or combination of DNA repair responses, basically.
What, if any, were the implications of this research, and how were they overcome?
The implication, yes, I think that it is a new protein in DNA damage response pathway, and intriguingly, I think it might be it has the potential to be also a Fanconi anemia gene and might be affected in this pathway as well, further downstream of this pathway. I should mention that we know now 15 genes which are involved in a Fanconi anemia disease. So 15 of them are mutated, causing the Fanconi anemia. But there are also some complementation groups of patients which we don't know the mutation yet, and hence don't know the gene which is mutated. So RNF113 might be also a Fanconi anemia gene, which acts downstream of FANCD2, because our genetic evidence is that it's parallel to the FANCD2, but further downstream on the event where homologous recombination repair happens, it might be involved there. So it's potentially a Fanconi anemia gene, but we also have to bear in mind that maybe also certain cancers might be defective in this repair and maybe accumulate DNA damage because of a defective RNF113.
Is there any room for future research following on from this paper, perhaps with the development of cancer drugs or treatments?
I think it's a very early stage now. We just have some description where it might play and what kind of response pathways RNF113 is involved. But I think the most important thing is now finding substrates for this enzyme and really further understand what kind of pathway it's playing with. I think this is an important next step. But in terms of drug, if you think about a lot of cancer drugs are actually damaging DNA. A very prominent one is called cisplatin. It's widely used in ovarian cancer treatment or testicular cancer treatments. And this drug also makes actually crosslinks. And what you can think about now is if you treat the cancer with this cisplatin, cells die. But if you make another deletion where you shut off a pathway which actually repairs this sort of lesion, you can sensitize these cells for the cancer drug cisplatin. So the idea is, for example, we can block now with a drug, for example, the catalytic activity of RNF113, which then should actually sensitize cells for treatment with interstrand crosslink inducing damaging agent like cisplatin. So you can think about combinatorial therapy, drug therapy, basically. And this is also what people investigate now these days a little bit to follow this up. So basically sensitize a cancer cell line, or some cancer cell lines might be intrinsically already more sensitive to crosslinks, and you can further sensitize them. Because for example, for cisplatin, there is a major problem of resistance. So these cancers respond very quickly to cisplatin. They die a lot, but after a while, the cancer is growing back because it's not sensitive anymore to cisplatin. But we can then maybe resensitize them by shutting off repair pathway, which are maybe upregulated in response to cisplatin. So there is definitely room there to explore, and several bigger pharma industry are focusing on nicotine signaling and try to target these pathways with drugs. But so far there's not really a drug out there which actually targets